Hello everyone, welcome to Unacademy. This is Ashwath. Today we'll be discussing about the meiosis stage of cell cycle and division. This is the second part of the video, where in the earlier part we have covered interphase and mitosis. So, in meiosis, reduction in chromosomal number takes place. So, the number of chromosomes are reduced in meiosis. If a cell has a diploid number of chromosomes, after meiosis it becomes haploid. Okay, so we can say that after meiosis only haploid gametes are formed, which include one set of chromosomes. Diploid include two set of chromosomes. Meiosis is of two stages, meiosis 1 and meiosis 2. In the meiosis 1, we can see the four stages: prophase 1, metaphase 1, anaphase 1 and telophase 1. In the meiosis 2, we can see prophase 2, metaphase 2, anaphase 2 and telophase 2. First we will see about meiosis 1 stage. The first stage of meiosis 1 is prophase 1. This is again divided into 5 subphases. The first phase is called as leptotene. In this leptotene, the nucleus increases in size and this absorbs water from the cytoplasm. As a result of this, these chromosomes are converted into chromomeres. The chromosome ends converge in one end of the nucleus and centromeres by the side. This leads to formation of a Boquet stage. Zygotene, the next phase of prophase 1. Here the chromosomes become shorter in length and they form pairs called as homologous pairs. This process of pairing is called as synapsis, where synaptonymal complexes are formed. So in zygotene, homologous pairs of chromosomes are formed by a process called as synapsis, which includes synaptonymal complexes. The next stage is called as pachytene. This homologous pairs forms tetrads. Okay. After formation of tetrads, a process called as crossing over occurs. Crossing overs only occurs between non-sister chromatids. This crossing over is nothing but the exchange of genetic material between homologous chromosomes. So in pachytene stage, the homologous pairs form tetrads. That means the two chromosomes of each forms a pair. Okay. So the crossing over occurs between non-sister chromatids and the crossing over is exchange of genetic material between homologous chromosomes. The next stage is called as diploidine stage. In this stage, at the site of crossing over, chromosomes get separated. The egg-shaped structure called as chiasmata or chiasmata is seen. Then the chromatids or chromatids condense and contract and become thickened. The next stage following diploidine is diakinesis. In the diakinesis, terminalization of chiasmata is seen. Chromosome fully get condensed and the spindle prepares separation of homologous chromosomes. So in this stage, the spindle starts preparing the separation of homologous chromosomes. During this stage, the nucleolus disappears and the nuclear membrane breaks. After this diakinesis process, the cell is ready for metaphase 1. Similar to the mitosis phase, in the metaphase 1, the chromosomes align themselves on the equatorial plate and the microtubule from the opposite poles simply attach to chromosomes at kinetochores. In the anaphase, the chromosomes separate where chromatids with centromeres remain. So during the metaphase, what happens? The spindle fibers attach to the centromeres and start pulling the chromosomes towards their poles. In the anaphase, what happens? The chromosomes separate and break, while the chromatids and centromeres will remain. In the telophase, the nuclear membrane nucleolus reappears, which was previously disappeared in the diakinesis stage. After the telophase 1, division of cytoplasm occurs. This is called as cytokinesis. There is a stage between telophase 1 and the next prophase 2. This phase is called as interkinesis stage. I am repeating again, there is a phase between telophase 1 and prophase 2. Between these two phases, there is a phase called interkinesis. There is a resting phase. After the completion of meiosis 1, the cell enters into the meiosis 2 stage. We have to note that in the earlier video we have studied about interphase stage of the cell cycle, where it has G1, G2 and S phases and G0 phases. So after the cell enters into S phase of the interphase, okay, it can go into mitotic cell divisions or it can come to meiosis cell divisions. So in the meiosis 1, the diploid cells will be present, they undergo division and they continue to meiosis 2 stage. In the meiosis 2, the first stage is called as prophase 2. After interkinesis, the nuclear membrane again disappears and the chromosomes become compact. Okay. The next stage is called as metaphase 2. This metaphase 2 is very similar to both meiosis 1 and mitosis. In this, the chromosomes align to the equator part or the center of the cell, and the microtubules from the opposite poles attach to kinetochores of the chromosomes. Kinetochore is a structure present on the centromere. Excuse me. Next, anaphase 2. The next phase of metaphase 2 is anaphase 2. In this, the splitting of centromere is seen, and after splitting of centromere of each chromosome, this move towards the opposite poles. Okay, by the end of anaphase, the chromosomes will be moved to the opposite poles of the cell. The telophase 2, the next phase of anaphase 2. Here the meiosis ends with telophase 2. In this, the nuclear envelope again appears and the division of cytoplasm is seen. It is called as cytokinesis. After the completion of telophase 2, finally from diploid cells, haploid cells are formed. Sorry, from diploid gametes, haploid gametes are formed. Here is a diagrammatic representation of the prophase 1 stages, which have 5 subphases. This is leptotene, zygotene and pachytene and diplotene. In the diplotene, we can see the crossing over. This area is called as chiasmata or chiasma. Their exchange of genetic material is seen in diplotene stage. And the second stage is diakinesis. After diakinesis, the cell enters into, or this enters into metaphase 1. In the metaphase 1, we know that the spindle fibers start attaching to the kinetochores or the centromeres of chromosomes, due to which they pull the cells towards the center of they pull the chromosome to the center of the cell. In the anaphase, the spindle fibers start pulling the chromosomes or chromatids towards their poles. As a result of this, cytokinesis occurs and divides the cell into two. Here the diploid stage is seen. Okay, diploid number of chromosomes is seen. After the meiosis 1, it undergoes into meiosis 2, where the same phases continues again, like prophase 2, metaphase 2, where the equatorial plate is formed, and in anaphase 2 they are started pulling towards the periphery, and finally they are pulled. In the telophase 2, again cytokinesis occurs and the haploid set of chromosomes can be seen in each cell. Okay, thank you. I hope you are clear with this concept. Thank you.